Hi all. Thanks for joining. In this short presentation, we will discuss how to build dihybrid Punnett squares and how we can use them to predict genotypes and phenotypes. Dihybrid Punnett squares are used to represent the genotypic and phenotypic ratios of two genes that are unlinked. As a refresher, unlinked genes are genes that are far apart from each other or on separate chromosomes and are therefore independently assorted when they undergo meiosis. Because we are combining two genes, this genetic cross is known as dihybrid. Let's take a moment to review some important concepts that will help us build and interpret our dihybrid Punnett square. Genes can have different versions, which we will call alleles. For example, hair can be brown, blonde, or red. The color of your hair will depend on what alleles for hair color your individual parents donated to you. The alleles can be either dominant or recessive. A dominant allele will mask a recessive allele. For example, let's assume the letter A denotes hair color. In hair color, the color brown is dominant over blonde. This means that if the allele for brown hair color is present, it will overpower the blonde allele, and this person will show brown hair. The combination of alleles from mom and dad are known as your genotype. Genotypes can be homozygous or heterozygous. A homozygous genotype means that the person inherited the same version of that gene, the same allele, from both parents. In our example, if one inherits a capital A from mom and a capital A from dad, the genotype will be capital A, capital A. Since they're the same, it's a homozygous genotype. The same would be true if one inherits two lowercase a's, by the way. However, if one inherits a capital A from one parent and a lowercase a from the other parent, then the genotype would be called heterozygous. The physical appearance that results from the genotype is known as the phenotype. In our example, since capital A denotes brown and brown is dominant over blonde, which is denoted by lowercase a person with a capital A genotype will have a brown phenotype. A person with a capital A lowercase genotype will also have a brown phenotype. Only the person with a lowercase a genotype will show the blonde phenotype. In order to make predictions regarding our genes, we need to carefully read and understand the information provided and what's being asked before attempting to perform the Punnett square exercise. Let's read the question and information in this example. If you cross a heterozygous red and short-stemmed rows with a heterozygous red and long-stemmed rows, what are the genotypic and phenotypic ratios? Red is dominant over white and short stem is dominant over long stem. We are being asked to cross a heterozygous red and short-stemmed rows with a heterozygous red and long-stemmed rows, and we're told that red is dominant over white and that the short stem is dominant over long stem. Given that information, what would be our parental genotypes? Let's do one at a time. The first parent is heterozygous for red, meaning it has a big R and a little r since red is dominant. This parent is also heterozygous for short stem, so it must have a big S and a little s since short is dominant. So the final genotype is big R, big S, little s. The second parent is heterozygous for red, so the genotype is big R, and it has a long stem, which means it must be little s, since we know that the short stem is dominant. So the genotype for the second parent is big R, little s. How do we determine the gamete combinations? We use the FOIL method. Because we are predicting the inheritance of two genes, each of which contains two alleles, we can have up to 4 potential combinations of gametes from one parent and another 4 from the other parent. An easy way to determine these combinations is using the FOIL method. FOIL is just an acronym to help you remember the combination order. The F stands for first, the O stands for outer, the I for inner, and the L for last. Let's take a look at how it works. You have a parent with a genotype big R, big S, little s, and you want to know the 4 potential gametes generated. Using the FOIL method, the first combination will come from the first allele from each of the 2 genes. In this example, it would be capital R and capital S. See the black arrow on the slide. The next letter in the FOIL is O, which stands for outer. So we would look at the outer letters of the genotype. In our example, that would be capital R and lowercase s. See the red arrow on the slide. The third letter in FOIL is the I, which stands for inner. See the green arrow on the slide. If we combine the 2 inner letters in our example, we get lowercase r and uppercase s. The last letter in FOIL is the L, which stands for last, for the last letter of each of the alleles for the 2 genes. In our case, that would be lowercase r and lowercase s. See the blue arrow on the slide. We use the same principle to determine the 4 gamete combinations for the second parent. Now that we know the 4 possible gamete combinations for each parent, we use them to help us predict the F1, or the progeny genotypes and phenotypes. Let's draw the Punnett square. It's going to have 16 boxes, and write down the 4 combinations for each parent as shown on the slide. Notice that the combinations for one parent go on the top, horizontally, and the ones for the second parent go on the side, vertically. Combine the corresponding parental gamete combinations to come up with the potential offspring genotypes. For each square, combine the gametes from the first parent and the gametes from the second parent that line up with that particular box. For example, for the first box, we take the information from the parental gametes in the first column and the first row. For the second box, in the first column, we use the information from the first column and second row, and so forth until we fill all of the boxes. Now that we've established all the potential genotypes that can arise from this set of parents, we want to report both the genotypic, letter combinations, as well as the phenotypic ratios. It is best, generally, to start with the genotypic ratios so that the phenotypic ratios are easier to establish. Simply count how many of the letter combinations within the 16 squares are the same. In our case, we have 2 big R, big S, little s, 4 big R, big S, little s, 2 big R, little s, 4 big R, little s, 2 little r, big S, little s, and 2 little r, little s. Make sure that when you add them up, you get a total of 16, equivalent to the amount of boxes that you created in your squares. We can also report that this is a 1:1:1:1 ratio, which is the reduced form. But regardless of which way we choose to report it in, we need to specify what genotype we expect in what proportion. What about the phenotype proportions? We know that red is dominant over white, and short stem is dominant over long stem. Using this and the genotypes we just wrote out, we get the following: 6 red with short stems, 6 red with long stems, 2 white with short stems, and 2 white with long stems, which can be reduced down to a 3:1:1 ratio. Thank you for watching, and have a great day.